Hello, hello. Okay. Hello. 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 Hello.
As Irrigation and CAD Department of Telangana. We have another guest, Dr. Apparao sir, who is we all know him. He's a very familiar face, who is the Vice Chancellor of University of Hyderabad. I request our director sir to welcome both the distinguished guests. Now I request our director, sir, to kindly initiate the proceeding for the day. Sir, please come on the dais.
Good afternoon, and it's a very great occasion for us. We are all here for the Science Day or Science Week celebration, frankly speaking. Government wanted us to celebrate Science Week. That makes us easy to celebrate our major program today on 27th. And we are really grateful, thankful to Rajas Kumar ji to take out some time for us. Professor Kodile, anytime we want, we fight out our time from him. So thank you very much to both of you for giving us time. And you'll be surprised that they both agreed to come on Sunday also. But then I thought that Saturday is also okay because government has paid us to celebrate the week. And this way it makes life easy. And I have a very pleasant thing to do. I will take 2-3 minutes to talk about Institute. If you can give the slide pointer. So Abhijit, so are you showing it to some kids also? Schools? Yeah. So there are a few schools which are joining. They will also see because this is our part of bridge program. So we do some work for the schools and colleges. So this is our institution which was inaugurated in this building in August 2018. Before that, we were in Miapur in a rented accommodation. Next slide. I can do it from here. So this is the building, sir, and everybody has now fascinated with the flowers and rainbows and natural things. So these are taken by some of the students. So now they have started doing this business. I have shown few, and then I think everybody wants. So this is not mine. Somebody has taken it. You change. So this is the inner courtyard. I just visited with the dignitaries, and ये कौन कर रहा है? तुम कर रहे हो कि मैं कर रहा हूँ? एक साथ में 2-3 जा रहा है. Okay. So this is in the evening how the institute's reception looks. And this is our boardroom where we have a lot of things. One of them was Indo-US meeting on biosecurity because we have the Animal Institute and now, you know, this is heating up. And we still are connected and I am a member of their Indo-USA interaction group. Next, okay. This is one part of the lab. Next. So mainly what we work on is mainly on animal nutrition, several infectious diseases, and diagnostics, vaccines for animals, animal reproduction. There's a lot of animals are infertile in our country. 35% buffaloes are infertile. So I think we are working towards increasing that, overcoming infertility. We have got a bioinformatics group and a center of bioinformatics where we have 4 bioinformaticians. So it's a pretty big, I would say, I have invested or risked to invest because out of the 19 faculty, 4 are working in one group. And I think that each of us in all fields, they need bioinformatics with big data nowadays for solving and resolving the issues in much easier manner. We also work on animal genetics and genomics, and we also work on transgenic technology. Especially, we are working on generating human therapeutic molecules, which are extremely costly, into the milk so that it can be sold at a much cheaper cost. So, next, Karo. So these are the various diseases which we work on. Next. And this is on the fertility. Next. And we are working on livestock genomics, mainly using the animal genetics, genomics, and bioinformatics, and also trying to really see paratuberculosis, differential immune response, heat resistance. So out of the chip which we have developed now, which in a month or 2 we will be going to release in the country, probably we can get various kinds of animals sorted out and to understand the genes which are helping them either to protect themselves or if there are genes which are allowing them to become susceptible to any disease. This is a COVID-related kit which is developed by Dr. Sonu Gandhi in our institute, and this has been given to an industry. And this is, we all know there is a disease called as brucellosis, which is causing around 28,000 crore rupees of worth of loss in country. And this can happen to human beings. It's a very big zoonotic disease. And we also know that there is inflammation of the cow's udder gland because of there's a huge loss of milk because of that infection and inflammation. So now we have developed a kit where easily in the field you can detect or kind of detect before the infection is shown as a clinical sign. Then it is very difficult to treat. So ahead of that, so preclinical mastitis can be caught. And Dr. Pankaj has done this work. Brucellosis is done by Dr. Girish Radhakrishnan. And there are, right, that several tests. We are also working on developing detection kits for venoms to differentiate which venom, if an animal is cut, so that what kind of treatment is to be given, which antibody is to be given. And this is what I talked about. We are generating in the udder gland. So I would simply say, I will jump here, that this is a experimented udder glands milk and non-experimented udder gland, non-lactating. And we have taken milk from lactating, non-treated udder gland. And what we find that we can generate human gamma interferon. This is the drug which is around 13,000 rupees one vial in this country. And a treatment of 3.5 months to 4 months is around 4-5 lakhs rupees. So we have now generated, this is a human kit brought from the market, and this is milk of mice. So in mice's udder gland, we have shown that we can generate human protein. Now we have got a big grant from Department of Biotechnology. So we are going to work with goats. Later on in future, we may work with the cow. So that reason is, if you want to have a factory, you have to have a license of the factory, space, manpower, 3 shifts working, electricity, everything. That cost will be reduced because if you generate it in the milk of an animal. It is possible. We have shown it. We have published 2 papers, and now we are working with the goat. So this is the farm which I have shown. This is the goats, and this is the animal house, small animal facility where we keep rats, rabbits, mice. So this is the inner corridor. It's a very state-of-art facility from inside, very modern facility. And next, these are the cows. We have seen it from outside, sir, just now. Next. Next. So we also go to various parts of countries and do a program called as Milan. So meeting of Indian livestock farmers and agriculturists with NIAP scientists. So this actually we try to find out what is the real problem of theirs, because most of my scientists, when they joined, they wanted to do whatever they have worked with. If they work with TB, they want to work with TB. If they work with any metabolic disorder, they want. So I tried to indirectly force them to go and interact and find out what is needed in the field, what the farmer wants. And we generated questionnaire on behalf of farmers. We gave mics to farmers, and sometimes some places said that we gave money to the people who organized. They said 2 lakhs rupees and 5,000 rupees honorarium from 3 professors to give lecture. Then we said, I'll give you 5,000 to 15,000 to not speak. Ask them to keep quiet. Let the farmers speak. Let us understand. And we kept 2-3 local veterinarians, converted that question into local language so that the veterinarian can convert it into English so that we understand what they are asking. We noted, we didn't answer a lot of things. And at the end, they were very happy that first time we could speak what we wanted to, rather than learning and listening without understanding what's going on. This program is going very good. We are going to make a compendium in form of a book very soon. Next. Next. So these are the programs of schools, girls' colleges, where we go and teach. Next. This is the same. They come to this auditorium sometimes, and then we bring them to lab and then bring back here, give some snacks, etc., and make them happy. Next. So these are the 3 major programs we are doing. One is the livestock genomics, where we are going to conserve our breeds. We have already made a chip. This is a very big achievement. Next. So this is one. Second is that we have now, after the COVID-19, we have really gotten 19 centers together, and medical colleges like All India Institute of Medical Science, Delhi, AIMS, Jodhpur, MGR University in Tamil Nadu, all big 7 medical colleges we got, and 7 big veterinary universities we got. NIB is the nodal center, and we want doctors and veterinarians to work together to address the zoonotic disease and how to stop the diseases at the level of the animals so that it never comes to human. So we will ask doctors also to take care, even if when they get the first or second report, they should immediately tell us so that we immediately, veterinarians rush, go with the state government in local area and try to confine the animal diseases and few human beings who are infected. So it doesn't spread like what happened in case of COVID-19. So this project is cleared by the apex body of DBT. This is a 32 crore grant. Probably in a month or 2, this will be released. Next. And this is how we have distributed work to various people throughout the country. Next for this project. Next. And this is the last thing where we just got a fund from Prime Minister's office, and we are going to develop a vaccine batch testing facility in this institution. So there are the three very big programs, I would say, which has given us honor, respect, and people at least know us that we are able to deliver few things and they believe on us. I will stop here. I will not go further from here. And with this, I will just introduce our dear Dr. Rajas Kumar. So you'll be surprised that he's PhD, and he is also done masters from Harvard University. I will just go through his profile. So he is a senior officer of the Indian Administrative Service, currently working as Principal Secretary to Government of Telangana in Irrigation, Environment, Science and Technology Department. In the past, he has worked in various policy making and policy implementing positions with the state and central government in India, as well as on independent consulting assignments in the United States of America and Europe. Dr. Kumar is an alumnus of Indian Institute of Technology, Varanasi, and holds a master's degree from Harvard University in Public Administration and International Development. He has earned a doctorate in economics from Osmania University, Hyderabad, where he stood first in the entire university. He is a recipient of various awards, including Asia Pacific Environmental Fellowship in 2003 and joined Japan World Bank Fellowship in 2005. He's an avid traveler and has visited over 25 countries in his official and personal capacity. And it appears that he is a great sportsman also because he plays golf, he loves hiking and reading. And it's our pleasure for having him here today. With this, I will request you, sir, to please come on dais and speak something for us.
Dr. Purile, Dr. Majumdar. That was a rather long introduction that he gave. But anyway, thank you very much for inviting me over. First of all, before I talk about my thoughts on what the National Science Day entails and what we should be doing on this day or in the days to come, I would like to very honestly admit that I'm delighted to be here. Dr. Majumdar's dynamism in making this place so alive and so beautifully kept is strikingly evident. And his dynamism was evident not only in what he has done here, but also in the manner he drove me around the campus and showed me everything. So thank you very much, sir, for a personal tour. Some of your issues which you have pointed out for water shortage, for soil availability and all that, I'll put the district authorities and as well as my irrigation people in touch with you. And the next time I visit, hope to see even like more growth here. Thank you. Again, you are doing some wonderful work here. I mean, the significance of this, I was not so fully aware. But some of them have immediate significance to the endeavors that we are taking up in Telangana. So as you are all aware, I mean, Science Day, you know, is a very important landmark, but unfortunately, I don't think that we take it with the kind of seriousness with which it is should be taken in the country. You know, our constitution talks, a directive principle says, what does it say? Who? Yeah. Somebody is murmuring. Hardeep Singh, aap to bol hi dijiyega. Main to student se pooch raha hu. Somebody? Who is that? Thoda khade hoke bolo yaar. Shabash. A hand de deta hai. Which article? Yeah, directive principles are there from 20 to 40 types, so it should be somewhere there. But, you know, it was the vision of the founding fathers of our constitution that scientific temper should come. I don't know, I sit amongst a community of scientists here, but as a district administrator, as a person who has been out in the field very often, it's shocking the kind of lack of scientific temper, the kind of irrational belief that people carry about today. And, you know, I mean, it really, really hurts the developmental efforts of a country. Nobody can afford less than us to have that kind of, you know, I mean, what shall I say, wrong notions about the reality of the world. In India, as you travel across the country and even if you travel in the neighborhood, you come across such superstitions and false beliefs and all that I don't understand where we have gone wrong. So my first request to all of you young people sitting here, including school students, I believe some of them are here. Not yet. Oh, they are connected. They are listening to this address. Okay. So to them in absentia, I mean through this virtual platform, my request is kindly, argue against, debate against irrational beliefs. What you understand to be the reality, please go and convince your brothers, sisters, the people in your offices, in your educational institutions, everywhere you encounter such wrong beliefs. And once that whole way of looking at things rationally, once the whole way of trying to decide an issue on the basis of reality as we best understand it today becomes a second nature to us, I think the country will progress also, and there all of us have a role as scientists, as citizens. Again, in my personal capacity, I think I'm quite enlightened to see the kind of work you are doing. As you are aware, Telangana has taken up first of all, it was already one of the more developed states in the area of, animal husbandry, farm stock breeding, the animal part of it. It was known as the egg capital of India, Nasik to Hyderabad, that road, many poultry farms and all have developed. We have taken up a massive, sheep distribution program in the state. I haven't handled that department, but I believe it has actually transformed the livestock scenario in the state. And the growth also is phenomenal. So taking some of your things, I see your photographs showing Prayagraj and Sambhalpur. I would be happier to see Mehboob Nagar, Jadcherla also. Remember,
So we first covered the country now.
sir, charity begins at home, sir.
We have started the program with VBRI, Veterinary Biological Research Institute of Telangana, and the University called UNIT. It's a big grant. It's around, I think, 20-25 crore grant we have submitted together for Telangana. We have also taken KP also.
Yeah. No, that's all right. Of course, like the gleanings and it's a national institute, and you are fully right. But I say that, there is a great need for your scientific findings, knowledge, and expertise to be distributed around here. I would suggest, now that I'm more familiar with your work, I have a few bodies under my administrative, control. The environmental EPTRI is under our biodiversity board is there. Secretary Animal Husbandry Department also I can call. They have very major programs. I don't think that the improvement aspects are given as much emphasis, or the current state of art understanding about animal well-being is brought into our policies as much as it should. I think your integration and participation in those things will be extremely beneficial to us. I would request that, and we also have you, sir, here. So kindly, I would personally, as the Secretary of Science and Technology Department, I would like to sort of involve you more into these programs. And there is tremendous scope here. I will not take much time. I think there is a keynote address from the Vice Chancellor. But once again, I would like to just thank you for inviting me over here and request you all to spread this message, not be in this function, passively attend and go. Just try and play your role beyond this also, beyond this week also. Thank you very much.
Thank you very much, sir. It's our pleasure to hear you. And also, as you understand that when you give little bit of support to them, so that when we interact, they will be more forwardly coming to us and interacting. Because several times when we go as a very they think we are a very hi-fi institution, going to kind of breach their territory. So it will help if it little bit of help comes from backside. But with great efforts in last 2 years, at least now I met the Vice Chancellor of the university here, Veterinary University, 4 days ago. And it happened that he listened. I was giving a keynote address to National Research Center on Meat. So whole country, there is only one center on meat, NRC Meat, Hyderabad. So they invited me for their foundation day lecture. And it happened that he was the guest of honor. So now he understood what kind of person I am. He had a lunch with me and he listened to my talk. So he was very impressed. And although we started Urjit program, but now he himself said we should be doing many programs like this. So I think gradually when a person interacts, relationship becomes and faith increases. So we are gradually succeeding in getting into the system and trying to collaborate with them. So that's what we are doing. Thank you, sir. Thanks for very good address. And I hope what he said, he will understand, school students at least, that we are scientists and we should spread science and realities. With this, I will just give a very brief introduction of Professor Rao, who has over 30 years of teaching and research experience. He is a senior professor. We all know he's a vice chancellor, a great molecular plant biologist, and he has several awards. I will not talk a lot. The list is very big, but everyone knows him, and I will not say that. He is elected fellow of all the major science academies of the country. He is J.C. Bose Fellow, Tata Innovation Fellow, K.C. Mehta Memorial Award for the crop protection from National Academy of Agricultural Sciences. He has chaired several important national programs, and he has guided 24 PhD students and 4 MPhil, and at present he has around 7 PhD students under him, and he has published about 110 papers in peer-reviewed, very high impact factor journal, and has about around 4,100 citations with a citation index of 37. H index of 37, which in scientific language, very high. So I will first convey my thanks to him for giving his time today. For us, it's a very important thing for us, and his time is very important. And with this, I will request Professor Rao to give his address. Thank you, sir.
Good afternoon, everybody. At the outset, I must express my thanks to Dr. Subir Majumdar, who has been my good friend, and probably almost a month back, he made sure that I will be available for this Science Day lecture. But I'm so happy to see Rajas ji amidst of us, a person who has, as a senior bureaucrat is one part of it that we heard, and so passionately attached to the science and the growth of science and the developments in science. That is something which we all should appreciate. I'm so happy, Rajas ji, that you are also with us today on a Science Day to share your thoughts. And when Subir called me, I was wondering, well, definitely I should not talk about the research that we do, and that's not the objective. Since it would be more a general one, I thought I'll make it a more popular one, the simplest possible, and maybe still drive a couple of points. The 2 points that I would like to drive is the matter ecosystem. That is the word that I would like you to take back. And the second important point that I would like to drive on a personal note, and just to tease Subir. Subir always thought that I am a plant science fellow, and why should we be collaborating with Apparao, and he will always be looking at animal biology colleagues in School of Life Sciences. And I will show him now, and to the scientists here, that there is a place where the plant scientists and this scientists working in NIAB can also find some collaborative work. So these are the two things that I will make in the next maybe 20-25 minutes. As I said, I wanted to make it as simple as possible. So many of you know that I work in soil microbiology and plant microbe interactions with very much closely associated with that in the last 4 or 5 years, more so on the soil microbiome and the root microbiome. And otherwise also we have been working more with the plant and microbe interaction. So I brought the livestock into this. Now, the day we celebrate the Science Day to commemorate the scientific contributions, outstanding scientific contributions that are made by Sir C.V. Raman. And I thought we should let us all pay tributes to this great scientist who has shown to the world that with minimum facilities, with not much of when he was in civil administration, and parallelly he was pursuing his science, and that's how he brought laurels to this country. And many people, he always felt that people should come to India rather than he going to other places. So let's pay tributes to this great scientist of the nation. And with this, I don't have to now repeat, but then I thought, these are the things that I have always noticed in the meetings that I associate with the NIAB, that the NIAB has been focusing on issues of animal genetics and genomics and transgenic technology, reproductive biotechnology, infectious diseases, bioinformatics and nutrition enrichment. Because several of these words are common. When you take out animal, the genetics and genomics is common for all of us. You take out the transgenic technology is common for all of us. Reproductive, you take out biology is common for us. Infectious diseases are common for us. So I find that there are many things that are overlapping in biology these days, and when we go to higher level, and that's what we would like, we can try to see one small point that where we have unification between the three components that I've taken: the soil, plants, and the livestock. This is another thing that has been fascinating me because it's linked to this bioinformatics components as well. The data, the amount of data that is growing at a rapid rate, and we see it. People estimate roughly around 10 zettabytes of data is existing today, and it is growing rapidly. By 2025, it may become 180 zettabytes. A lot of our sequence data information, whether it is the genomics sequences or proteomics sequences, metabolomics that we are talking of, all of them are getting deposited. They are also huge. When Subir was mentioning about a core group in NIAB engaged in bioinformatics, and every institute, it has become essential for us to have strong groups in bioinformatics, and more so at University of Hyderabad. You know, we have a computational and systems biology department I have created in the last, very recently, 3 years ago. I have elevated that center to a department and brought good people to engage in this area of research. So the data, more and more data that is being generated, what we are doing is we are trying to take out some information out of it quickly, but we are not taking complete information out of it. Sometimes it becomes essential for us to see how this voluminous data that we are generating, how do we make use of it, and what are the multiple ways, other than the ways in which we have already explored, what are the ways that we can have alternate algorithms and then can retrieve the information, hidden information in this voluminous data. For you, three slides were jumping, and for me, not even one is moving. Yeah, right. These are the, I just start because there may be some audience, virtual audience, who may be very new to science or just at the beginning of science. I thought I'll introduce a small thing about microorganisms and their ubiquitous nature. They're everywhere. These microorganisms are everywhere. And you talk about the ice, you talk about the ocean, you talk about the soil, you talk about the plants, you talk about your livestock. They are there. They can be good, they can also be bad, and for some, we don't know what they do. There is a major issue for microbiology even today, but that doesn't seem to be a major issue, although a large, huge chunk of these microorganisms, we cannot bring them onto culture. But we are at least bothered now. When we realized, we were worried, what would all those 99% of organisms or 99.5% of organisms which we are not able to culture, what they must be doing, we were worried. But more and more of this metagenomics, when it has come into picture, we now understand even their functions, how these unculturable organisms may be contributing to the ecosystem in which they are present, we are able to understand. So there are several issues that they relate around environment, around the organisms themselves. And this question people were wondering, can we culture the unculturable? Well, some of them we are able to culture now. We have even our own lab and different laboratories have cultured. But that, as I said, is not going to make a big difference whether we are able to culture all of them or not. If we can culture it, yes, we can study better. If we cannot culture, there are ways and means of studying them. This is the word that we commonly use, the word genome, which is common to all living organisms on Earth. For everything, there is a genome. Whether you call it as a bacterium, you call it as a plant, you call it animal, for everything you have a genome. And interestingly, all of them have universal language and an instruction manual. That genome serves as an instruction manual for that organism that we all understand about. And it is a toolbox where you have a lot of things built in it. And it's also a record of history. If we take this as the broad outline of the genome, as I said, this talk cannot be like a scientific talk talking about genomics. That will be boring for the children who may be attached, and some of you who are not into genomics also will find it boring. And also, if I talk about the microbiome that we are doing, that also will be boring. I thought I will avoid all that. And this is something which has come in the past 2 decades. It has become more popular, this metagenomics, where a collection of these microbial genomes or the genome fragments directly extract from the environment and then we study them. And that's with the power of the sequencing tools that have become available, now we are able to do at ease this metagenomics, and in no time and with less money. When it started, we were spending lakhs of rupees on sequencing alone, and now we are spending less than few thousands for sequencing large amount of DNA that you have, provided you have your own methods of designing the primers and then preparing your samples and pooling them in the right way, and you have all the jugglery associated with it. So this will help us in many ways. This has been helping us in many ways. The kind of questions that people have been asking on this metagenomics: who are there? What are all the organisms around? That metagenomics answers us. And what are they doing? What exactly are these organisms doing? We have not cultured them, but we still are able to talk about, because we have all the backup from the genomics data that is available, and metabolomics data that is available, and metabolic pathways that we have deciphered, everything that is helping us. And how are they doing it is also able to explain. We are able to partly explain, making use of the same data that we have got. So this has made the life relatively comfortable for us to have a quick glance of what's happening in a given environment. And then an interplay of organisms, like what I have started off, the plants, the soil, and the livestock. The interplay. I'm just reminded of one small thing here. When I was a PhD student, we used to send some cultures for identification. Those days we were sending to a scientist by name Bradbury in United Kingdom, and it's long ago I'm talking about, and he would we are confident that we have isolated it from plant, and then when he sends me an identification, it shows Enterobacter cloacae. Wait, we have isolated, where is this Enterobacter cloacae coming on the plant? We were never able to follow. But today we know all that, okay? So you look at, I'm not here to complicate the things for you. I'm only interested to show livestock is not different from agriculture. In Indian agriculture system, everything is together. The soil, plants, and livestock are together. You cannot separate them out and then say that I'm only dealing with this. Well, at the level of growing the animal, milking it out, and then making use of it is different versus when you look at scientifically, they are interlinked. And when you look at the kind of microbial groups that are associated, they're not different. The proportions of the microorganisms that are associated might vary, but then the groups of organisms remain the same. And if we go into more details of it, obviously we will find where from those differences have come, how those have come. Things have to come from soil to the plant and from plant to the animal. All this has been happening. The connections, this is what I'm trying to say, connection between the soil, plants, and animal microbiomes could be the meta ecosystem that I'm talking about for us to look at closely. So the soil has rich microorganisms and the plant has microbiomes, the soil microbiomes. Some of them get specialized and then you have only certain number of them associated with the plants from this soil. And then from these, the ruminants will be acquiring their microbiomes. You look at the interesting thing that happens. I'm trying to see a plant root, whatever it does, the human gut also does the same thing, absorbing the nutrients. Okay, that is and you also see a spatial separation of microbial components both on the surface of the root and also in the gut. There is a spatial separation of groups of microorganisms, and you will find some similarities again in terms of where it is, in terms of pH, in terms of oxygen, in terms of different kinds of things that are around it. And those are the things that are common between these two that we have to understand. And the bovine gut microbiome is no different. Okay, the functions of the bovine gut microbiome I was trying to understand when I come. It improves the feed efficiency, maintenance of host immune functions. Rumen methanogenic bacteria contribute to the methanogenesis, ruminal methanogenesis, and augment production of methane and other greenhouse gases, and contribute to waste nitrate production as components of feces and urine. These are certain functions of bovine gut microbiota. These are coming from the microbiome rather than from the animal. Sorry, this time it jumped. And people now show that there are heritable phenotypes, that the rumen and gut microbiomes as heritable phenotypes also has been shown, at least to some extent now. So there is a mutualistic, commensal, and parasitic microorganisms that reside in the rumen and the lower gastrointestinal tract of cattle and other ruminants exert enormous influence over animal physiology and performance. This we are not familiar with. Thanks to the human microbiome and the dimensions in which it has grown and the contributions that we know how the microbiome is contributing to the human health. Well, there is I'm not saying that there is not similar studies are not done here, but maybe there is more scope for us to do. the bovine milk microbiome originates from gut via enteromammary pathway is something which has been shown in 2016. So the milk microbiome originates from the gut via this enteromammary pathway. So these are the things that indicate to us that how microbiome can be influential and how the interacting microbiomes can be important in studying these components together and understanding it as a we cannot separate it out now, the soil, plant, and the livestock. The connecting link for all of them now being the microbiome. This is the point I wanted to drive here. I would skip these couple of slides. Yeah, the mastitis, I was looking at that slide. Now you can see the milk microbiome composition is an important determinant of mammalian health, plays an important role in other health by interacting with the immune and metabolic functions of the cow, the spread of the virulence factors, and the spread of antimicrobial resistance genes. Here we can see the variation in the components of the microbiome in different stages of related to the mastitis. The healthy animal has a different proportion. The other different clinical mastitis and different things have different microbiome proportions. So this also can be an influential factor. I'm sure the people here in the host pathogen interactions are very closely monitoring some of these things in their own studies. With this, I wanted to say that microbiome and animal health are very intricately associated, and the relation between these microbial communities will lead to new methods of diagnosis, treating, and also prevention of the diseases. This is some methodology that normally people follow. And I feel this interaction studies, meta ecosystem studies, if we focus upon, they can help us to discover new enzymes, antibiotics, and other reagents which are not known so far, and we can look at more exotic habitats. The habitat we have to define. What is that habitat that we have to we want to look in? That's what we have to define. And improved tools will quicken analysis. This has been happening, and I don't know what kind of new things will come in the years to come. And it also helps us investigating ancient DNA remnants. This also we have witnessed in the last 10 to 12 years. That's where I said it's a history. The genome has a history, brings a history. Discoveries such as phylogenetic tags, these are general things for the microorganisms. Learning about the novel pathways will lead to knowledge about the current and non-culturable bacteria, to then learning to the culture these systems. That also will be helpful. I think with this, I am closing my whatever brief lecture that I was expected from Subir Majumdar on this day. And I would like to greet all of you once again on a Science Day, remembering the contributions of a greatest scientist like C.V. Raman. And again, reminding that I'm so happy that it coincided, and Subir made it sure that Rajat ji is with us, and it made my day. I'm so happy to be here with all of you. I must express my thanks to some of the slides that you have seen towards, particularly towards the end. These are brought to me by one of my students, Mr. Anirban Basu. He is not a doctor. He's a PhD student. And of course, I would like to thank him, although he's my good friend, Subir Majumdar, for inviting me here. And also all of you here sitting, and also those who are elsewhere as virtual audience. Thank you very much.
Thank you, sir. Thank you. And it's a great honor to have you here. And as he keeps on saying, that we are very good friends, because we are not only neighbors, but we are kind of the same kind of human beings, and we like each other very much. And he has helped us in growing very well, because he is in most of our governing body. He never misses. He's in our finance committee. He's in our society. So he has been closely watching our progress, and he is very happy with our progress, and that's why he supports us everywhere. And his words are valued because he is vice chancellor of the topmost university of the country. He's not just Dr. Appar of Bovine. His management abilities and the way he has progressed the university is really very well done. And I thank Rajas because somebody who could have been present here today, the best person would have been who is in charge of science and technology of this state. And as he said rightly, that we are in this state, and I think my scientists have to learn from what he has said, which we have been telling and forcing them, that work for the place where your situation is situated. That's what Dr. Ashwadan is telling now, that wherever you are located, ensure that you work for them. And it's a great pleasure, sir, that you could come here and you could see what kind of science we do. And if you find anyone under your umbrella who can be benefited by us, or even our collaborative mutual benefit can come to both of us, I think you ensure that you direct them to us. And we'll be more than willing to really go fast forward and interact. That we have started now. We took some time to settle in this building. We were in rented house. And I think now we are running on track, and we are ready to take on board anyone who wants to join. So 18 to 22 years it took in between COVID went. That didn't stop us. So we are moving, and now we are capable of giving space to people who can have a ride with us in the field of biotechnology. This is, I give a small token of respect for you. I cannot dare to give you honor of your So with this, we thank all the students and faculties and fellows, postdoctoral fellows, and thank you for joining us. I hope you had tea and snacks before coming here. Okay, take care.